The overall goal of these procedures is to visualize and track the progression of somatic cell reprogramming into induced pluripotent stem cells, or iPSC.
This method can be used to answer key questions in the iPSC field, such as how reprogramming kinetics and reprogramming efficiencies are altered by the somatic tissues used, the reprogramming technologies applied, and the matrix and media combinations used within. The main advantage of this method is that it utilizes cell surface markers to look at the transitions of somatic cells into iPSCs, such that you could use this technology to be able to monitor in real time the reprogramming events.
On day 7 post-transduction, wash the fibroblasts with 2 milliliters of DPBS per well and add 0.5 milliliters of 37 degrees Celsius 0.05% trypsin EDTA to each well for a 3 to 5 minute incubation at 37 degrees Celsius. When the cells have rounded, stop the reaction with 2 milliliters of 37 degrees Celsius fibroblast medium per well and gently pipette the medium across the bottom of each well to dislodge the cells. Transfer the fibroblasts from each well into individual 15 milliliter conical tubes and collect them by centrifugation. Resuspend the pellets in 2 milliliters of fresh 37 degrees Celsius fibroblast medium and count the cells from each culture condition. Next, seed 1 × 10⁵ cells into pre-prepared feeder-independent conditioned wells in a 6-well tissue culture plate and incubate the transduced cells in the cell culture incubator overnight. The next day, replace the medium in each well with the appropriate medium for the cell application.